Hi, I'm Sean.
Hi, I'm Charmin, and this is our presentation on the proteomic analysis of protease-resistant proteins in the diabetic rat kidney. The aim of our presentation is to show how Western blotting contributed to the identification of the protease-resistant proteins from the streptozotocin-induced diabetic rat kidney.
First, a few key definitions. A metabolic disorder that causes excessive thirst and production of large amounts of urine.
The process of bonding between sugar molecules such as fructose or glucose without the controlling action of an enzyme.
A kidney disease which is very common in diabetic patients.
The directed digestion of proteins by protease or intramolecular digestion.
The process of passing fluid through blood vessels or the lymphatic system. The branch of genetics that studies the full set of proteins encoded by a genome.
As we said previously, the experiment looks to identify the protease-resistant proteins in the kidney of a diabetic rat, specifically those involved in glucose metabolism and stress response.
The addition of ribose sugar to a protein.
Rats were injected with nicotine amide followed by an injection of streptozotocin, 65 mg per kilogram of body mass, dissolved in 10 millimolar sodium citrate buffer at pH 4.
After 15 days, samples of the kidney were extracted. The experimental procedures used upon these samples were enzyme assay. These are laboratory methods that are used to measure enzymatic activity in the experiment. 7 different enzyme assays had been carried out. Protease activity assay,
Gel protease assay,
in-gel protease assay, proteasomal assay,
proteasomal assay,
superoxide dismutase assay,
lactate dehydrogenase assay,
alcohol dehydrogenase assay, and glutathione S-transferase assay.
peroxide dismutase assay, alcohol dehydrogenase assay, and glutathione S-transferase assay.
The specificity of the antibody-antigen interaction enables a target protein to be identified in the midst of a complex protein mixture.
Blotting was carried out twice, pre and post-trypsin digestion. Generally in such experiments, two classes of antibodies are used, monoclonal antibodies or polyclonal antibodies. These primary antibodies incubated the different membranes at 37 degrees Celsius for 1 hour. Western blots or immunoblots checked a specific protein immobilized on a membrane. Proteins are separated by SDS page, in the case of this experiment 12% SDS page, and transferred from the gel to a hydrophobic or positively charged membrane. The membrane uses polyvinylidene fluoride membrane, and this was left incubated at 4 degrees Celsius in a blocking buffer containing 5% skim milk powder dissolved in tris buffered saline.
This had been done to reduce non-specific binding of the antibody. The blots were washed with tri-buffered saline, which contained 0.05% tween 20, once and twice with tris-buffered saline only. The membrane was then incubated for 30 minutes at room temperature, with a secondary antibody conjugated with biotin conjugate. This will allow for the bound antibody to be detectable in a variety of ways, which mainly relies on the secondary antibodies that bind to the primary antibodies in this experiment.
In this experiment, the secondary antibody was linked to the enzyme streptavidin conjugated horseradish peroxidase. This produces a colored fluorescent band after being incubated diaminobenzidine. Semi-quantitative reverse transcriptase PCR. The combination of the physical separation abilities of liquid chromatography and the mass analysis capabilities of mass spectrometry. This is a method known for very high sensitivity and selectivity.
The other method used was Western blotting. Blotting refers to the transfer of biological samples from a gel to a membrane and their subsequent detection on the surface of the membrane.
The image here shows the result of blotting. In lane 1, the marker can be found. The SDS page marker positions the molecular weight sizes on the blot. Lanes 2, 3, and 4 are trypsin digestion. Lane 2 consists of bovine serum albumin only. Here there is a heavy band. In lanes 3 and 4 there is a faint band in line with the heavy band of lane 2. Lanes 5, 6, and 7 are post-trypsin digestion. Like lane 2, lane 5 consists of bovine serum albumin only. Bands bands can be seen separated, but no band is present on the same weighting as seen in lanes 2, 3 and 4. The same can be seen in lanes 6 and 7. No bands are present at the weighting of BSA.
The plots show that glycation and ribosylation induced protein aggregation and resistance to trypsin digestion are compared to unmodified BSA. These results suggested that glycation-induced PRBs can be identified by this approach. Furthermore, the feasibility of these in vitro results were demonstrated in complex protein mixtures. The image here shows another set of results of blotting. The blots were used for age and ubiquitin modifications. In lanes 2 and 3 are the controls. Lane 2 holds the lysate control and lane 3 holds the diabetic control. These are both pre-trypsin digestion.
The plots show that glycation and ribosylation induce protein aggregation and resistance to trypsin digestion as compared to unmodified BSA. These results suggested that glycation-induced PRPs can be identified by this approach. Furthermore, the feasibility of these in vitro results was demonstrated in complex protein mixtures. The image here shows another set of results of blotting. The blots were used for AGE and ubiquitin modifications. In lanes 2 and 3 are the controls. Lane 2 holds the lysate control and lane 3 holds the diabetic control. These are both pre-trypsin digestion. Lane 4 and 5 contain the post-digestion samples. Figure A shows the protease-resistant proteins. Figure B shows the samples with anti-AGE. Figure C shows the samples with anti-ubiquitin. The diabetic kidney protein shows several protease-resistant bands, whereas the controls do not. These results were supported by previous studies where proteins such as collagen were shown to be protease-resistant in diabetic conditions. Under normal circumstances, misfolded protein structures and potentially harmful protein aggregates are ubiquitinated and removed by the proteasomal system. This reinforces that glycation or ribosylation induce protein aggregation and resistance to trypsin digestion as compared to unmodified BSA. However, there seem to be more ubiquitinated proteins in the diabetic rat kidney than in the control group. The protease-resistant fragments in the diabetic rat kidney were also found to be ubiquitinated. After the detection of ubiquitinated PRPs, protease and proteasomal activity were investigated. Only uses semi-quantitative reverse transcriptase PCR revealed upregulation of GAPDH, HSP60, SOD, GST, LDH, and ADG at a protein and mRNA level in diabetic rat kidneys. Upregulation is an increase in the number of receptors on the surface of target cells, making the cells more sensitive to a hormone or another agent.
Overall, the experiment carried out showed decreased proteasomal activity and a decrease in the removal of these proteins, which contributed to their stability and may lead to cross-linking and aggregation. The results which were obtained provided an idea to the possible cause of protease resistance and the consequences which lead to diabetic complications.
Looking at this experiment, Western blotting contributed to identifying the protease-resistant proteins. The first blot showed the effects of glycation and ribosylation, while the second blot highlighted the presence of PRPs with AGE and ubiquitin modification. With the addition of the other procedures, the paper concluded that there was a decrease in proteasomal activity and removal of these proteins.